to work with a different kind of cells. They call HEC293 cells. They are very used in the whole community of researchers, investigators. They are used for expressing different kind of proteins in the plasma membrane. So this is very nice because this lab that I'm doing a collaboration here at UFSC, they express at BK channels in these cells. BK channels are potassium channels that are dependent on intracellular calcium. So this is very nice because since I was doing this collaboration, here are the HEC293 cells, I decided to try my hormone there. So I added reverse T3 in this system, and very quick, like 1 minute, RT3 activated the BK channels of the cells expressing HEC293. I mean, the HEC293 expressing BK channels. And then I wash out the hormone and I block it with the potassium blocker in the end of the experiment. So what I showed here in expressed BK channel cells, it was very interesting because corroborated with the data I got in Sertoli cells. Besides this collaboration, I had another collaboration with another professor that works with Sertoli cells, that she works with ATP. ATP usually binds to the P2X7 receptors. These receptors are ionotropic, so once the ATP binds to the P2X7 receptors, there is an influx of positive charges inside of the cell that could be sodium, calcium or potassium, especially calcium. And then it was very interesting because I found that ATP not only activates the efflux of potassium, which is a washout and then block it, but also ATP is so potent that I was able to measure the depolarization effect of ATP. So here I have the resting membrane potential of Sertoli cells, minus 50 millivolts. Whenever I added the ATP 1 millimolar, I found a huge depolarization in the presence of ATP, and when I washed it, the cells, the effect was gone. Why this is important? Because this is the first step of the RT3 effect. First, RT3 activates a depolarization that we don't really know if it's calcium or sodium. We believe it's calcium, but after that the cell is super depolarized, and what happens? The repolarization of the cells, which I measured here with the efflux of potassium from these cells. So this should come first, depolarization, and then here repolarization of these cells activated by ATP. So this is very nice because this professor, this student actually, it's a PhD student, she's showing that ATP is very toxic for the cells and activates a lot of kinds of oxidative stress and things like that. So this is another project we're running in the lab. And also I added two other compounds in the cells, which is very interesting, but I didn't bring it today because it's exactly the same data. It causes a huge depolarization of the cells and the flux of potassium, which is ethanol. Ethanol is very toxic, as we know. So this project is very interesting because the student is giving ethanol for the animals in the lab, and we can see that it's very bad for the development of the mice in the lab. So this is just showing in vitro what it was shown in vivo, that not only ATP but ethanol and also Roundup is very toxic for the cells too, and Roundup, as much as ethanol, causes a depolarization. I mean, a depolarization showed like this, and after that a repolarization of the cells. So this is what you have with Sertoli cells. And then another project that we're running in the lab is potassium channels in beta cells from pancreatic islets. So just to remind you first, when you have glucose entering inside of the cell, you have the increase of ATP concentration. ATP is going to close the voltage dependent, the ATP dependent potassium channels. Then we're going to have a depolarization, and it's going to open the voltage dependent calcium channels. The calcium is going to activate the insulin release, and then in the end of that mechanism we are going to have the efflux of potassium. That it is what we actually are doing in the lab right now. We are measuring the efflux of potassium. That comes after all this mechanism here: entrance of glucose, close of potassium ATP channels, opening of calcium dependent voltage channels insulin release, and then you are going to have the efflux of potassium. Here is just to illustrate what happens in the membrane, in the action potentials in beta cells. First we have the influx of calcium and then the efflux of potassium, which means depolarization, repolarization, depolarization, repolarization, depolarization, repolarization. So I think it's clear. This is the data that I have to show you here today. It's a master degree student. She showed that a new hybrid that's derived from glibenclamide, it decreases hyperglycemia in vivo, and it has a very low index, insulinogenic index. So it appears this new hybrid coming from glibenclamide, a derivative from glibenclamide, it's potential drug in the future for treating type 2 diabetes, because it has less side effects. Apparently it has less side effects than glibenclamide. So I have an islet here, and first of all I showed that the islet was responding to glucose, high levels of glucose, because high levels of glucose activates beta cells. So just to show that it was beta cells, I first added glucose to show that this depolarization and the flux of potassium causing by glucose that activates insulin secretion. And then I added this new hybrid, this new compound to the cell, and I found that there was a efflux of potassium or repolarization of the cell, indicating that the influx of calcium that was measured by this student with the radio-labeled 45 calcium, I was able to reproduce here the repolarization of the cells showed by the efflux of potassium. This has nothing to do with what I was talking about here, because this is diabetes, and I just switched to this slide, but here it's very interesting. Here it's another collaboration that I have been doing here in the university, where we are working with H4060 carcinoma cells from human lung, and this professor, she wanted to see if the sodium channels are localized in the plasma membrane of the carcinoma cells that she is culturing in the lab. So she gave me these cells, and I found out in the literature that this channel was already, the sodium channel, voltage dependent sodium channel, was already characterized in these H46 cells. So I took all the protocols from this paper, I took all the same solutions, and I reproduced the data in the cells that this professor gave to me, and I was able to record the sodium voltage dependent sodium channels exactly the way that he did in his paper. But the news here is that we have a new hybrid that calls JAV. This hybrid, it's very interesting because it blocks the sodium potassium pump. It's known to block the sodium potassium pump, and here we use it in voltage dependent sodium channels, and you kind of saw the reverse of the sodium ions from the outside of the cell to the inside of the cell. And we are still studying why this is happening, but we believe it's very important, because what happens with the sodium channels is that they are involved in metastasis. So it's very well known in the literature now that sodium channels are one of the main candidates to be involved in metastasis. And since we put this new compound, and you saw that we have an inhibition of the sodium currents, that means that this new molecule could be inhibiting metastasis in this kind of cancer. So this is a very nice project as well. And now I would like to talk a little bit about calcium, about intracellular calcium. What happens in intercellular calcium? Calcium imaging. So calcium imaging, I usually use the FUTA2. FUTA2 is a calcium indicator. It's fluorescence. So whenever you have an increase of calcium inside of the cell, FUTA2 is going to bind to calcium, and you're going to have a fluorescence, a high fluorescence, which is going to be a pulse, indicating that you have an increase in intracellular calcium. For example, 3 millimeters, this is an islet, a human islet. For example, in low concentrations of glucose, we don't have increase in intracellular calcium. However, in 16 millimolar glucose, we do have an increase in intracellular calcium to trigger the release of insulin. So we have here the fluorescence, because we have the FURA2 binding to calcium, and that's the way we measure calcium. That's why we call calcium imaging, because you can see the imaging here of when you have the increase of intracellular calcium. So now I'm going to show two kind of data that I have done with calcium imaging. It was not in Brazil, but we would like to set up the system to work here at UFSC as well. We have all the equipments, it's just the camera that is missing. But now I'm going to show you some data. I work in this lab in Diabetes Research Institute in the Miller School of Medicine, Florida. They showed for the first time that the human islets are different from the human mice. And they found that this unique cyto architecture of the human pancreatic islets has implications for the islet cell function, especially the paracrine effects. The paracrine effects between or among beta and alpha and delta cells, you can see that here they are going to talk to each other much more than the mouse islets. So this is something important. And this lab, they used to study the physiological functions of neurotransmitters that are localized in the human islet. So they showed many kind of work. They showed that acetylcholine is involved in insulin secretion. They showed that glutamate is involved in autocrine effects for glucagon release. They showed that ATP-gated P2X3 receptors also are involved in the insulin secretion. And that's when I start working with GABA. And I started working with a different technique, which is very interesting, because this technique with calcium imaging, it allows us to see what's that the release of GABA from the human islets can be detected by these cells that we actually acquired, these cells from the Novartis company. And these cells are very interesting because these cells, they are sensitive only to GABA. So these cells, the carpet of cells, the CHO cells, they are sensitive only to GABA. So when I make a control with these cells without islet, I don't have any kind of signal here. There is no intracellular calcium or anything. However, when I add exogenous GABA to the system, I can see a pulse. That means that the extracellular GABA that I added in my system is binding to the GABA receptors in the CHO cells, which were incubated with FURA2. That's why the CHO cells are going to be measured in the presence of FURA2 to see the increase of intracellular calcium. And then when you have the GABA binding to the GABA receptor here, it's going to activate the protein G, and this protein G is going to activate the increase of calcium so we can measure. Look, in the presence of islets, when I put the islets on the top of these biosensor cells that are sensitive only to GABA, I found out for the first time that the GABA release from the human islets are constitutive and rhythmic, the release of the GABA in this system. So this is what happened when you put the human islet on the top of the biosensors. They start releasing GABA without any kind of stimulus, and then the biosensors can sense the GABA from the human islets, and they show you that this GABA has been released just as insulin is released every 4 or 10 minutes. It was a very interesting finding. And when I tried in human, monkey and mice islets, I found that actually the GABA secretion are conserved among all the species. It doesn't matter if it's human, monkey or mice. The GABA is always being released, and then you want to know how this could be important for insulin release. So we have the access to human islets from donors, and these human islets, they could be sometimes be type 2 diabetic. So when you have the access to type 2 diabetic islets, we would try here to put them on the top of biosensors, and it was very interesting because the diabetic type 2 was not able to detect any kind of GABA coming from the islets. So we start thinking that GABA is important because there is no GABA in type 2 diabetic patients. The controls we did for that, we did a control with KCL in the end of the experiment to show that the islet was alive, was viable, right, in the presence of KCL, and then this is what should